Hello,
Hey,
everyone.
Dorcas.
Hi, Mark.
How are you doing?
I am well, thank you. And thank you for accepting to present this meeting today. Welcome.
It's okay.
Hi,
From
Elisa, Paula, Randy, Thiago.
Hello, Dorcas.
just so you know, this meeting spans 2 days for me. So it's just coming up towards midnight for me. 11 p.m.
Sorry about that. But anyway, we are excited to hear from you. So it
Okay.
should be a couple of minutes before several other colleagues join. Oh, wow. Just checking the chat here. Sometimes there was also a link to Teams. Some people go to Teams instead of yeah.
Ah, okay.
Yeah. Wow, sorry to keep you up all this late, but the only
That's okay. I went to
catch.
sleep and set an alarm and woke myself up again, so it's okay.
Yeah, all right. No problem.
So while you're waiting for everybody, is this a topic that you as a group have been discussing, or was there a specific reason that you were interested in me actually discussing this topic?
Okay, so it is a topic of interest to most of the teams that will be attending today. It's part of our accelerated breeding work plan on innovation and improvement and optimization, of course, what we have been working on. It's one of the things that we are working to improve through the crop teams that will be represented here today. And so, yeah, we would like to listen to other thought leaders in this sphere. And so we're honored with your presence today.
Okay, thanks.
Yeah. So it's just gonna be about 3 minutes more, and then we will start when
No problems for me.
turn. Yeah. Sorry.
Just while we're waiting, in about 4 or 5 days, I'll be flying to Nairobi. So I'll actually be in Nairobi next week. I don't know if anybody told you that, Dorcas, but there's a BPAT review that's going to be taking place in Kenya.
Yeah. Yes, we are looking forward to meeting you in Nairobi, actually, because
Okay, good. So I'll get a chance to meet you there. Good.
Yes. We had spoken with Chris, and Prasanna also reached out yesterday. So we'll be joining some of the sessions while you are here in Nairobi. Yeah.
Really good.
Yeah. So looking forward to. Hi, everyone. Welcome. So we'll give it 2 more minutes, and then we'll start as other colleagues join. So I hope no one is lost. Yeah, go ahead, Mark.
I was just going to say, I just sent you a copy of the PowerPoint. So you have the slides that I'll talk to. You feel free to share those around with your colleagues, and we can
Thank you.
certainly use those as a discussion point in Kenya also when I'm there. So look forward to that.
Thank you. Thank you very much, Mark. Oh, people are lost in the Teams link. I thought I had...
Teams, so.
Oh, sorry about that. I thought I had deleted that with sending out this invitation, a reminder for this month, but it seems it's still a problem. I don't know if it's possible to delete that from the links team without deleting the Zoom link anyway. So, yeah. But welcome back. I hope everyone has found their way back to here now, hopefully. Okay. All right, I think it's 3 minutes past and it's time for us to start as other colleagues join. So it's my pleasure to welcome everyone here today. For those joining us the first time here, this is a COP meeting that is for breeders, CGR NARES breeders, that is actually hosted currently by the CGR's Accelerated Breeding Initiative. Previously, we have hosted it as EIB Module 2. And in this COP meeting, we have invited talks and discussions around topics relevant to breeding optimization and modernization. And it is a platform for all of us to share information and seek clarification over those topics of interest as they pertain to activities in our own programs. And so normally all questions are welcome. And today, today's speaker is Professor Mark Cooper. And thank you very much, Mark, for agreeing to present today, even though it's about midnight, your time. Professor Cooper is the Deputy Director for the ARC Center of Excellence for Plant Success in Nature and Agriculture. His main research interests combine theoretical simulation and experimental investigations to formalize the use of prior knowledge of trait genome to phenome architecture to design breeding strategies that can deliver accelerated crop genetic improvement for quantitative traits. He has breeding experience in both academic and private sector, and he has kindly accepted to speak to us today, and we're looking forward to that. So, Mark, I'll pass the floor over to you.
Thanks very much, Dorcas, and also thanks everybody for joining. So I really appreciate the invitation. So I'm at everybody's mercy for you to get the most out of this. So Dorcas mentioned, as some of you were coming on, that this is a topic of interest to you. So I would encourage you to ask questions and get the most out of this rather than me just giving a talk. Usually with these things online, it's a little bit sterile because you can't actually see people. But hopefully, Dorcas, you can keep track of if questions come up or if things need a little bit more discussion or clarification. Please sort of try and get my attention through the chat or something, and I'll try and do my best to address questions. So I'm going to share my window with the PowerPoint, and if Dorcas, if you can sort of give me a confirmation if that's actually working okay, I'll put it on full screen.
Yeah.
You can see that okay?
We can see the screen, Mark, thanks.
Okay. So Dorcas gave me a title, and I only made one change to it. So the title, I guess, also has quite a lot of specifics and jargon within it, so that I guess I'm assuming that a lot of people are very familiar with this topic of target population of environments and the idea of representative testing strategies and breeding programs, and how. And so Dorcas said how the views on this topic have evolved over time. It's probably a bit bigger than I could do in a single presentation, but I could certainly address some of how my views on this topic have actually evolved over time. So what I'm going to do is try and give you my perspective with a bit of a historical context of, I think, some of these ideas. And one of the things I just wanted to start off with, and I'll put a laser pointer on. And so Dorcas, again, can I just check? Can you actually see the laser pointer moving around on the screen?
Yes, yeah.
Okay,
You
good.
can see.
Right, yeah, excellent, thank you. So I sort of just did a screen grab of the cover page of this publication I was involved in that was actually published back in 1996. So it seems like a long time ago now when you actually think about that that work. But this sort of, a lot of the ideas that sort of from the perspective that I've seen, we were sort of asking the same question that you're asking right now, back in the 1980s and 1990s. And this publication actually came out of a relatively small workshop that we actually ran from a number of colleagues around the globe that were working on this topic, in partnership with CIMMYT, IRI, ICRISAT, and ICARDA at the time. So a lot of these discussions were actually being conducted through collaborative partnerships with a number of the international centers. So individuals from within those centers that were working on this topic, we actually came together for about a week to give a whole series of presentations and have discussions at the ICRISAT headquarters in Hyderabad. And out of that, a number of the presentations went on to become chapters inside this book. So a lot of the origins of the ideas sort of that I'll try and cover were in play in the 1980s and 1990s. And this sort of perspective of how they've evolved is has grown out of the research that was conducted following some of the directions that we started to form inside the publications that you can find inside this historical book now. The one big shift that I think that I've certainly seen, and what I've tried to be doing in this work, is that the whole topic of trying to deal with genotype by environment interaction in plant breeding has sort of got its own interesting history, but it really sort of is an afterthought if you think about a lot of the way people think about breeding, and it's something that you try to pay attention to after you've dealt with genetic variation or broad adaptation in a breeding program. Whereas for me, it never was sort of a secondary thing. It was actually pretty core to how you might think about designing a breeding program. So instead of thinking about it as G by E, it was sort of, you know, what is the interplay between genetics and environment? And to be able to answer some of the questions people were asking, you need to have a better understanding of what is this environment that you're actually trying to understand. And that sort of provides a bit of a foundation for the target population of environments. But instead of thinking about that external to breeding, what I've certainly seen there's a lot of interest in doing is bringing this to be internal inside the core principles of designing the actual breeding program. And so what I've just sort of written down here in the bottom left is what I think is the most appropriate, simplest form of the breeder's equation for a plant breeder rather than an animal breeder. And it's got some very explicit definition of terminology inside the breeder's equation that isn't typically there. And I'll try and explain why I think this explicit form is much more useful if you're thinking about design and optimization of testing strategies within the context of a breeding program. And that is, there's some of the things that you're familiar with, but there's an explicit term in here that I'll spend obviously a bit more time discussing, and that's the genetic correlation between the information we can get out of the multi-environment trial and any other trait measurements that we're actually taking, and the expectation of that genetic variation inside the target population of environments. So I'll expand on this, but what I'm really trying to argue is that we want to internalize the thinking of the target population of environments explicitly as part of the breeder's equation, not something we do outside of the breeder's equation. And a lot of the discussions that I've had with people, that the consideration of the TPE and trying to define that as something that's done outside of the design of the breeding program and the breeder's equation. And so a bit of the outline of how I'm hoping I'll be able to get to doing a better job of explaining this extended form of the breeder's equation compared to the way that it's often written down is I'll sort of just do a little bit of a outline of the theoretical framework that a lot of people might be familiar with when they think about the breeder's equation, and just think about its origins to where I guess I'm proposing we should be focused a bit more with this extended form of the breeder's equation. It's extended only in the sense that it's more explicitly defined, and it's often defined when people write down the breeder's equation. And to do that, that sort of provides the framework for talking about the target population of environments, moving from a lot of the early discussion of this topic, which, you know, there's some really landmark publications. Most people, I'm sure, on this video conference would be familiar with the publication by Comstock and Moll at a conference that was held in the US by the National Academy of Sciences and National Science Foundation, looking at issues of quantitative genetics and applications to breeding, and this was within plants and animals. So it's sort of a really interesting publication to actually read today, and think about how the people were tackling this type of question back in the 1950s and 60s. I think the first explicit use of the term target population of environments that I found in the literature as a set of words, even though I think a lot of people were thinking about this before, but the use of this TPE terminology, I think, first came as a set of words in a publication by Comstock in 1977, which was in the first International Conference on Quantitative Genetics. Sort of an interesting publication as well. And the thing that was interesting about it for me was that it wasn't even a key talk that was given. It was just one of the contributed talks, and there's this terminology here. Interesting for me is Comstock went on to publish a book on quantitative genetics and design of breeding programs that I think came out in 1996 with Iowa State University Press, and he didn't really talk much more about this. So a lot of the consideration really came down to thinking about how do you sample locations in years without really thinking about much about the biophysical properties of the environment. So there was not a lot of really detailed understanding of G by E interaction, but there certainly was a consideration of a sampling perspective. So that within that, there was always a lot of discussion of topics such as how many locations, how many years, can you substitute years by sampling more locations? And you know, a lot of these types of ideas were sort of present in the literature. You can see a lot of publications that talk a lot about locations versus years, how many of each of those. And I think that lends itself pretty straightforward to statistical analysis, and you can find a lot of publications on that. And I just see that I got a typo here, but there wasn't really much consideration beyond the numbers to thinking about the types of environments that you were actually sampling in locations and years. But then a lot of people started to look a little bit more at studying the specific details of genotype by environment interaction, rather than just sampling years and locations. So there was a lot of pattern analysis work, clustering, hierarchical grouping of environments to try and find groups of environments, for example, as targets. think out of this, you can start to see the emergence of people thinking about, as well as the numbers that you sample, what is the alignment between maybe the target population environments you're thinking about and the multi-environment trial, how well it aligns with this target population environments. But a lot of this was really quite external to this theoretical framework of the breeder's equation that actually sort of started with some of the publications that you'll see from Lush and Falconer back in the 30s right through the 1950s. And so I won't dive into that in too much detail, but I'll talk a little bit about that. I think that now that we're a little bit more comfortable talking about MET and multi-environment trial and TPE alignment, a lot of the ideas around applied phenomics, genomics, and enviromics that is a terminology that sort of has appeared pretty recently. Along with these, it seems everybody wants to have an omic something or other. But yeah, for me, a lot of the things people are doing in this space can actually be focused around key questions that we're asking in breeding around this MET TPE alignment. But what I'll try and do to help the discussion is just sort of point to a few examples for me that I've touched on over the years from when I was involved in wheat breeding in Australia from the 1980s to 2000. And 2000 basically because I left Australia to go to the US at that time, but I've sort of maintained some interaction with people that were working on these wheat breeding questions starting back at this time. And also a bit more about some of the maize breeding work that I've been involved in. And I saw that Peter Coldrake and Randy Hawley are actually on this talk as well. So I think yeah, they could also add, if they wanted to, some of their perspectives on how this is sort of evolved also over time within some of the applications of maize breeding. And I think beyond just thinking about genotype by environment interaction within maize breeding, the sort of biophysical environment, we can think about agronomic management in combination with the sampling of the environment and think about this whole G by E by M space and how we can improve yield stability for crop performance in a target population environments and also multiple crops in a cropping system. A lot of those ideas have sort of taken shape from the work I started with what was Pioneer Hybrid at that time in the 1990s, joining them full-time in 2000, being involved with the development of these types of technologies as a maize breeder within Pioneer. So that's sort of the overall perspective and structure that I'll try and follow. So if anything's a bit confusing, please just jump in and ask, you know, how this relates to that overall structure. Thought it was really good, given that Dorcas asked me to talk about how this has evolved. I think one of the things that's always been interesting to me is that if you actually look at how the way people write down the breeder's equation, the basic structure of the breeder's equation was really written down originally by animal geneticists and animal breeders, not plant breeders. And so it actually has a flavor that actually has its origins in animal breeding. And Jay Lush from Iowa State University is often given sort of credit as being the first person to actually talk about the breeder's equation in a formal way. I sort of have got a copy of his original publications, and if you actually go into his book on one of the early editions, page 140, he has this sort of picture that sort of thinks about truncation selection within an animal population. And then he talks about the idea of how do you realize genetic improvement over cycles of breeding, where you're applying a selection differential. So he sort of nicely defines that in his book, but you don't realize all of that. So there's this heritability term, and he sort of has this terminology in here that really focuses on the additive components of the genetic variation and thinks about the total variation, also considering some of the non-additivity, dominance and epistasis. But the thing that's sort of really interesting to me is there's no consideration of G by E interaction throughout this whole book. So as an animal breeder, this wasn't something that he focused on. And so the structure of this breeder's equation really didn't give a lot of consideration to, how do we think about genotype by environment interaction in the structure of the breeder's equation. So I think that sort of influences the way people think about things. The other set of really interesting publications in the history for me, and if you go back to Falconer's original publication of Introduction to Quantitative Genetics, which was published first in 1960, is sort of an interesting publication prior to that I'm highlighting here in 1952, where Falconer, as an animal quantitative geneticist working on mice and all these types of organisms, and interacted with a number of really important animal quantitative geneticists that I'll highlight, had this nice publication in 1952 where he actually talked a bit more explicitly about the breeder's equation, or the form of the breeder's equation, taking into account genotype by environment interaction. There's a nice path analysis type diagram where he said we're really interested in this from a correlated response to selection perspective, where you could think of selecting in one environment, what is the correlated response in another environment? So you could think about the expression of the phenotypes in environment 1 and environment 2. There's a correlation, but you could think about these as two independent traits, and the realization of this phenotype is a function of the macro environmental components, the partition of the phenotype into the genetic variation and the microenvironmental variation within those two environments. And there's a genetic correlation, a consistency or lack of consistency between the two environments, the correlation of the genetic component, and there could be a correlation of the environmental terms, the microenvironmental terms, but says with appropriate experimental design, this should not exist. And so what you're then interested in is decomposing this phenotypic correlation into the genetic correlation. It's going to be a function of the square roots of the heritabilities in both environments. So there's a fair bit of nice discussion there. And even prior to his book in 1960, you can find a form of the breeder's equation that's written down as a direct response equation here, where you got the response as a function of the selection intensity, the square root of the heritability, standard deviation of the genetic variance, or as an indirect response version of the equation, where you're looking at response in one environment based on selection in another environment. So you've got heritability 2 here, genetic correlation, then the genetic variation back to this target environment. This sort of then carries into Falconer's book in 1960. And again, you have an explicit version of the direct response to selection breeder's equation here. The interesting thing, even though there were no molecular markers or people were thinking about genomic selection at this time, Falconer actually does give a lot of the reforming of the breeder's equation from being expressed in terms of standard deviations of the observable phenotypic variation into an explicit form where you're looking at the standard deviation of the additive variance, or the variance of the breeding values, and the square root of the heritability, which is what people often think about as the predictive accuracy in the genomic selection terminology that is often used when you see this form of the breeder's equation used in combination with different breeding strategies using genomic prediction. And at that time, he actually made it pretty explicit that this is a useful form of the breeder's equation when you're making comparisons of different methods of selection. So a lot of that sort of is pretty well understood from a quantitative genetics perspective back by 1960. And this, even before we had molecular markers, you could actually see this framework that we use today forming at that point. The other thing that's interesting to me is, and this is sort of the origins of the way that I've thought about internalizing the considerations of the target population environments into this breeder's equation is that in Falconer's original book, he has his correlated response to selection equation. And so this to me is the origins of this extended form of the breeder's equation that I'm much more comfortable using. And you can actually see the equation that I had on the first slide, correlated response, written here, where he's got Y, but I put target population environments. His X for me is, I replaced X with M for multi-environment trial. And you can actually see this explicit form written here as sort of a correlated response where you're thinking about character X to character Y, but this same framework can be extended to thinking about multi-environment trials as a sample of the target population environment. So these origins go right back to this work by Falconer that's pretty well captured in 1950s and 60s. The thing that's also really worthwhile pointing out, because Falconer wasn't really a strong researcher in this space, so I sort of have spent a bit of time speaking to others about this that were familiar with Falconer's work at the University of Edinburgh, and speaking to Bruce Walsh, who a number of you might know. He says that probably a lot of this work was explained to Falconer by Alan Robertson, and you can actually read in the acknowledgements of Falconer's book that he credits Alan Robertson with a sort of a major part of helping with the writing of this book. So it's sort of a pretty interesting historical perspective. And if you're familiar with Alan Robertson's work, he did a lot of work on this genetic correlation and the sampling variance of this genetic correlation that I'm talking about. Okay, so it's sort of a bit of the foundations. For me, around the time of this publication that I mentioned at the beginning, I was really interested in the fact that it seemed to me there were these two groups of people doing analysis of G by E interactions. There was an analysis of variance perspective. There was a lot of work on cluster analysis, grouping, principal component analysis of environments within the types of data sets that we can create in breeding programs, where you've got a whole range of genotypes from 1 to thousands, if you want, tested over from a few environments to hundreds of environments. And we get these matrices of data that we're trying to analyze with these different methods. And so what I've just sort of really highlighted in here, there are all these ways that are being applied to analyze the data set. And this publication that I was involved in just really tried to show that, well, all of these methods really are just interrelated, and you can actually show the interrelationships between all of these methods. But ultimately, what you're trying to do is understand the genetic variation that resides in here to make predictions back to a set of environments that you're sampling. And instead of a single target population environments, you're probably dealing with a mixture of different environment types that you're encountering inside this target population environment. So I was really interested in how we were sampling these, how we would characterize them as a mixture of environments representing the target population environments. What were their frequencies of occurrence? And then I sort of looked at the basic form of this prediction equation here, the breeder's equation, and then if you extend it to deal with G by E interaction, the interesting thing that you realize is that all of this G by E interaction is really dealt with as error. So it's in the denominator. So it's not really accessible as such in a selection program as a direct target in breeding. So even if we talk about a target population of environments and G by E interaction, there's not an explicit definition of this in the numerator, even though it could be there because of the way we sample things. It's not explicitly defined, so you're not really targeting it in your breeding program. But the correlated response form does give you an explicit way of thinking about this. And so this then, if you start to think about redefining the breeder's equation in terms of the correlated response framework, you can actually start to put some of the aspects of the adaptation to different environment types, the G and the G by E, into the numerator with this framework. And so the problem that sort of got me sort of really into this in a lot of detail was this early work that I was involved in, which actually started off as my honors project and became my PhD project at the University of Queensland. Was as a wheat breeder in Australia, we were using germplasm from the CIMMYT program quite heavily in the northeastern region of Australia. They were producing a lot of data in their international testing program. That germplasm was working really well in our target environments. So there was obviously some correlated response from CIMMYT's breeding work and their international testing program into the environments that were relevant in our northeastern region of Australia. So I was trying to understand how that germplasm was being developed by CIMMYT and why their breeding strategies was giving realized response in our target environments, and how we could use this to accelerate and target the movement of germplasm from the CIMMYT program into the Australian breeding programs. And there's some early publications that I just highlight there that were sort of what I was looking at with CIMMYT as part of my PhD project. And then subsequently, after I finished all that, in collaboration with other people at CIMMYT, Kai Matthews led some work to say, well, this was sort of really interesting, but maybe a few sort of years later, so instead of the germplasm that was being developed in the 1990s, what was happening in the 2000s, and found that this sort of relationship was pretty repeatable. The genetic correlations were repeatable across germplasm across years. So there were things in common between probably the genetic variation we were selecting for in international testing and some of the requirements for performance in northeastern Australia. So, starting to try and understand that better. And that sort of then drove work led by Karen Chenu and Scott Chapman and others to try and do a better job of characterizing what are the main environment types in terms of water availability in northeastern Australia. So thinking about looking at water availability and different levels of water stress, and trying to separate the environments in terms of environments where there was a limited level of stress. So this ratio here, I guess for our purposes of discussion, 1 means that there's very little stress, 0 means there's severe stress. This is looking at overdevelopment from planting through to final harvest of the crop cycle. If the line stays near the top, there's limited amounts, mild levels of water stress. If the line goes down, you get into more severe stress during flowering time. Some of that could be relieved by rainfall during the grain filling period. Some of it could persist, so you get a very strong terminal stress. These became sort of the 3 key environment types that we were targeting for wheat breeding in northeastern Australia from the drought adaptation perspective. And so throughout this region that we were breeding for, we started to break that down into regions and look at the different frequencies with which these different environment types actually occur. So we're starting to form a basis around which you could start to characterize the target population environments in terms of the environment types, their frequencies of occurrence, and then start to think about the genetic variation that's required for performance in these different environment types. So that sort of takes me to maybe a little bit of an example with an empirical data set to actually show this, where what I've really had was an experiment that was run over about a 15-year period, where I actually had 2 sets of experiments I had with common genotypes in them. And one of these was what I would call my multi-environment trial at specific locations we were using in the breeding program. And then there were a lot of on-farm sampling of the same, experiments in the on-farm conditions using the management strategies that the farmers were using. So that became a representation of the target population of environments. And so I had these 2 data sets, common genotypes. So here's a sample of those genotypes. And if you just took the average performance of these genotypes across the multi-environment trial data set and compared that to the TPE, that was the sort of relationship you had. And to give you an example of how this fitted into the breeding program, this group of germplasm here was pretty representative of what we were sorting out inside the Australian breeding program at the time. These three individuals here are representative of some of the new germplasm that was coming in from CIMMYT that we were trying to learn how to use to improve yield, but also the quality of this wasn't good enough, so we had to improve yield and quality simultaneously. So we're trying to take advantage of this yield opportunity, but not lose the quality that was required for the wheat varieties in the northeastern region. Applying the work of, so this picture back here of these different environment types, what I've done is I've just organized this data set, looking at all the environments that went into the multi-environment trial data set, the TPE data set, and just broken this out into two groups of environment types. Environment type 1 is the mild stress, environment type 2 is all of the drought stress. And if you look at the relationship for the same genotypes for yield between the within the multi-environment trial, between environment type 1 and 2, that's a sort of weak relationship that we actually saw. If you did the same thing in the TPE, this is the sort of relationship you saw. If you compared between the MET and the TPE data set, the relationship for yield of the mild stress, there's pretty good relationship. For the more severe stress, there's no relationship. So this sort of then underpins the sampling of the environment types in the breeding trials versus the on-farm type situation in this case. And then you can start to look at what happens in terms of the genetic correlation between what you see in your multi-environment trial and the target population environments as you sample different frequencies of environment types. So I'll just look at three quick pictures. One, this is, if you ever look at a review article of G/E interactions, people often emphasize this crossover interaction. All I'm sort of pointing out is in my wheat experiments, if you pick two of these genotypes, genotype 3 and genotype 8, and you looked at them in terms of two of the environments, we saw a lot of crossover interaction that we were trying to sort out as the G/E interaction. If you were to take this classical picture and then say, let's compare environment type, the multi-environment trials with the TPE and change the frequency of these two environment types in your trials versus the target population environments, depending on how you sample these environments in your multi-environment trial, you can have anywhere from a positive to a negative genetic correlation to performance in the target population environments, depending on the frequencies with which these environment types exist in the target population environments. So if you do a bad job of aligning your multi-environment testing system to the TPE, you can make great progress in your data sets in the multi-environment trials, no matter how big they are, but you can go backwards in the target population environment. So that's a direct implication of this crossover interaction and the frequencies of occurrence in the multi-environment trial and the TPE. So you want to start to think about this relationship more explicitly within the design of the breeding program. We can sort of say, well, that might be a bit easier if we found this new genotype, genotype 12, that was better than everything else that you were studying before, like in the data set. But it's still not quite that simple, because you can actually still get, if you see here for these three genotypes, it doesn't solve the problem. You don't always have a positive correlation if you're sort of undersampling certain types of environments from the TPE that are important in the TPE inside your multi-environment trial. So again, you can easily have good statistics in your multi-environment trial, but poor correlation to performance in the TPE. And obviously, where you'd like to be is spending a lot of time up on the top of this response surface and making sure that your multi-environment trial is well aligned with the requirements of the target population environments. If we take this full data set now And I said that the overall correlation on average performance was about 0.7 with the target population environment. So we just averaged everything over the 15 years. This would sort of, one possible sample of environment types in the multi-environment trial relative to the TPE. And if you looked at the full response surface, this is sort of what it actually looked more like. If you think about the frequencies of the environment types in the multi-environment trial and the frequencies in the target population environments. And the way that we were sampling initially was probably more sitting down here. And so this required a bit of a redesign of the multi-environment testing system to push it up onto this sort of higher genetic correlation with the target population environments by redesigning the way that we were actually sampling environments inside the breeding testing program. So we were starting to pay a lot more explicit attention to this genetic correlation by characterizing the target population environments and then redesigning the multi-environment trial so this becomes an explicit part of what we're looking at. So that sort of is a wheat example, just really quickly, just to finish off. The same principles were applied in the maize breeding world. So the framework is directly applicable to any crop and any system. If you start to think about the framework, we were trying to understand the G by E interaction for the US Corn Belt. And then we sort of use that to build a picture of the target population environments, not unlike the paper with Karen Chenu, but we were, instead of thinking about it just in terms of 3 or 4 environment types, we were sort of interested in the whole continuum of water availability. And then the question was, how well could we design testing systems to sample the range of water availabilities that we were actually targeting in the US Corn Belt. And so in this situation, we were able to find a number of locations where we could actually design multi-environment testing within managed environments that did a good job of stratifying the sampling of the range of water environments and exposing a lot of the G by E interaction associated with that range of water availability to target selection that allowed us to think about drought adaptation as one of the traits that we were selecting for as part of a breeding program. And just sort of putting a few pictures around this. So this is an example of the early stage testing part of my breeding program at one of the stations where we have different managed environments to sample this range of water availability to try and represent the different types of drought environments that can occur throughout the Corn Belt whenever water limitation is a factor in the target population environment. So this is just a large experiment that was run by the business unit testing products across the US Corn Belt. Each point is a location. A green point just means that it was more of a favorable environment. So 800 millimeters plus, or a red environment is one where water limitation was a factor. So you've sort of got this mixture of drought and favorable environments. Then in 2012, it was relatively easy to sample those because of the widespread drought that occurred. Then you can start to look at testing your germplasm across this continuum and thinking about making sure that you weight the information from these environments to take into account the genetic correlation in your testing system into the target population environments. So you can now start to explicitly think about how to design your multi-environment trial to represent this target population environments. The other piece that I'll just highlight down here is that for drought performance, we did a whole lot of work to try and understand some of the traits that were driving that. So here's an example where Charlie Messina was actually looking at a series of 9 traits and the interactions of those traits. And over this continuum of water availability, he was looking at how the different traits contributing to performance in drought contributed to the yield variation in the different water environments in these experiments and in the target population environments. The interesting thing is you can see at the extremes that there are some dominant traits that are important, but they're actually different in the favorable environments than in the water limited environments. So in terms of the breeder's equation now, you can think about the design of your multi-environment trial to represent your TPE, to explicitly think about the genetic correlation. You can actually think about the trait variation that's contributing to your yield variation in each of these environment types across this continuum. And you can actually think about how that genetic variation is expressed in your experiments, how what you're seeing in your experiments actually relates to the TPE, and you can actually start to think about that in terms of any of the quantitative genetics frameworks and trait frameworks to try and explain the yield variation in these different environment types. And all of this sort of is a bit easier to think about in terms of this extended framework here. And this sort of then just gave me as a breeder a bit of a sort of a framework for thinking about when I'm putting selection pressure on yield potential, when am I really selecting explicitly for drought tolerance, and when am I selecting for yield stability around this whole continuum of water environments. Rather than just selecting for drought tolerance, we wanted to select for products that would push this whole front of performance up, or remove some of the instabilities from the products that were crashing because they had weaknesses in some of these water limited environments. So we could start to select for traits that were driving yield variation in these environments. So this there's a summary of some of the history of this work with some experimental results by Charlie Messina in Crop Science, published in 2022, where he actually shows how over cycles of breeding, these fronts were moved forward over the different generations of some of the products that came out of these breeding programs. So it then shows you how you can take this whole idea and put it into practice inside a breeding program and actually drive towards your yield stability targets explicitly by thinking about the traits that are contributing to variation across this whole continuum. So it then maybe this is the slide that really is the single slide that answers Dorcas's question to me. How has this evolved over the years? I think that one of the things that I found in discussions with people is that there's a lot of work that's done on, for example, genomic prediction, where we're interested in improving predictive accuracy by getting some large sample of data in a multi-environment trial. And we think about comparing different ways of improving predictive accuracy and optimization around this equation. The thing that's really interesting about this equation is that it is the extended form of the breeder's equation, but I'll put it to you that it explicitly states that the genetic correlation between what I see in my multi-environment trial and the target population environments is 1. That's why you don't see it in here. So it's assumed to be 1, whereas in most data sets I've analyzed, it never is 1. So, if you focus on only using the data that you've sampled without understanding how these data relate to the target population environments, depending on how your multi-environment trial has sampled the continuum of environments, you're going to expose genetic variation from different traits, and you're going to optimize to different parts of this whole continuity of performance that you're ultimately interested in. So if you oversample the favorable environments, you'll optimize your selection strategy towards the types of traits that contribute to improved performance in these environments, but they may not be well suited for some of these stress environments. And I found that in both wheat and maize in different geographies. So this is pretty much what was playing out inside the breeding programs that I was working within. So I think that what I'm much more comfortable in working with is an equation that explicitly forces me to ask the question, what is that genetic correlation between what I see in my data sets and the target population environments, and making that an internal discussion about always trying to think about how well my multi-environment trials are actually representing the different views I have of the target population environments, so that I can understand the genetic variation that I'm exposing in these multi-environment trials and its relevance to these target population environments. So what it sort of then means is that the equation is almost the same, but instead of actually thinking about just the genetic variation you see in the multi-environment trial as being explicitly the variation in target population environments, this component here is actually replaced as the product of the genetic correlation between the MET and the TPE and the genetic variation that you're ultimately really interested in, and that is the variation that plays out on farm in the target population environments. So these equations are really the same. It's just the breeder's equation that you actually often see people writing is this equation, but you assume that this is one. There's maybe no misalignment between your multi-environment trial and the TPE, and therefore if you make this one, then these two equations are exactly the same. So I would say that, you know, even though people don't realize it often, they're making an explicit assumption that there's a genetic correlation of one between the data sets they've got and the on-farm target population environments. And if you ever spend a lot of time explicitly trying to estimate this, that's often not the case. And so that sort of would be what I think is a really interesting discussion to have about how to think about characterizing the TPE and sort of internalizing this within the optimization of breeding strategies, explicitly taking into account this sort of component of the breeder's equation. And I've just shown you a couple of examples of how I actually did that. So I'll sort of stop there, and hopefully that was maybe a bit useful, and I'll stop sharing my screen now, and hopefully, Dorcas, that was on target of some of the topic that you wanted to talk about or not. Then you can ask questions that are different if you want to.
Thank you very much, Mark. That was a nice presentation. So I think we do have a couple of minutes to ask questions and have a discussion around this. So I'll open the floor. If you have any question, please just raise your hand. Yes, Gary, please go ahead.
Hi. Yeah, thanks, Mark.
Hey, Gary.
How you doing?
Good.
This correlation between the evaluation environment and the farmers' production environment is the great unknown of most plant breeding programs. So it's remarkable that we don't estimate it. It's particularly important for us to understand it in the context of agricultural systems that haven't undergone an agricultural intensification or transformation, because and so it's particularly important in sub-Saharan Africa. The conditions on farms in sub-Saharan Africa for the great majority of farmers still are extremely different from conditions on the research station. In most cases, they're different in terms of fertility, they're different in terms of plant density, they're different in terms of competition from intercrop. And even if we try to mimic those conditions in trials on the station, we're not really going to, we run a danger of missing the target. So, it's really important for CG programs that are focused on, particularly on that type of system, to understand that correlation. And it doesn't mean we have to shift all testing on farm, but it does mean as we move into genomic prediction that we need to have estimates of performance on farm when we train our models. So we have research starting in maize and in beans where we're putting stage 1 trials for model training purposes through tricot type of designs on farm under farmer management. so that we're able to train the model on a trait performance under farmer management in these systems. It's really important for us to be doing that in all of our systems. So, you know, for example, the CIMMYT wheat program exerts an immense amount of selection pressure at one location at Obregon. So it's critically important that that program understand the genetic correlation between performance in the environments at Obregon and in each of the TPEs. So it's an you know, thanks for highlighting it. It's a you know, and there is a long history of estimating that and discussing that correlation, but it's never really been adopted by plant breeders as an important parameter that they have to understand in optimizing their programs. And now, you know, we really have to do it if we want to make progress in difficult environments.
Yeah, no, thanks for adding to those comments, Gary. Totally agree. I mean, if I look at my whole career, it's sort of pretty sad sometimes to sort of condense it down to one thing, but I think, you know, one of the things that I've spent a lot of time working on with people is trying to understand what that correlation is for the way that they're actually doing testing. And the unfortunate thing is when you ever do estimate it, it's always often way below 1. And it sort of then starts to make you realize a lot of the tense discussions that have been had over the years between the way breeders are breeding and not achieving the realized gains that they would anticipate on farm, exactly as you said. I think that goes, I mean, Salvatore Ciccarelli was a participant in that workshop that I talked about that we held at ICRISAT that gave rise to that book. And he was really pushing for this on-farm participatory plant breeding as a strategy to try and deal with this same type of issue back then. And you know, I think it's taking a long time to actually shift people to think about this. And I think my biggest concern is that I spend a lot of time with people looking at genomic prediction strategies that are not very familiar with sort of understanding on-farm performance. And so they get these data sets and they pretty much spend a lot of time doing cross-validation within those without even thinking about how relevant the data sets actually are. And so, you know, I like the genomic prediction approach. I've used it a lot myself. It works really well, but you better make sure you're working with a data set that's relevant for your targets. I think you and I have had a lot of good discussions about this over the years.
Okay, yeah. Thank you, Mark. Thank you, Gary. Eduardo, you have your hand raised. Please go.
Yeah, I would like to go in the same lines than Gary, because at the end of the day, I don't think I get some of the answers that Mark, you can give us. As Gary was mentioning, and you were emphasizing, that is the important value, the correlation between the MET and the on-farm trials, let's say, or the on-farm targets. So we're moving to what Gary is describing as some sort of breeding on the farm. Based on GS to make sure that that correlation is maximized. But of course, most of the programs across the CG will have this uncertainty whether that is going to be better than more of a classical approach, you know, just sampling, let's say, a couple of locations, let's think for a minute on 10, and assuming that those are highly correlated with the on-farm. So you presented at the end some of the results from this high scale or big scale MET. And I have the feeling that probably you already have some of those answers as well. What is in your experience, or what have you observed? You know, how many is the classical approach still, you know, powerful? And if that is, how many locations? You know, we need to really, you know, sample. I guess that depends on how complex the TPE is. Or do you think is, you know, a good idea what we're planning and Gary was describing as this, you know, on-farm GS?
Yeah. No, I think it's going to take a fair bit of rethinking about not only how many we sample, but getting the right measurements of learning a bit more about how well we're sampling the continuum of conditions. And I mean, I think the framework tells us we've got to pay a lot more attention to this and think about how to do it, Eduardo. I mean, even within the maize data sets that I studied a lot of the historical data sets in the pioneer maize breeding programs. And what we actually found was that, I mean, I'll boil it down to a couple of key points. The reason we could accelerate breeding for the stability of performance across the norm of reaction for maize to develop these Aquamax hybrids was because there was genetic variation for these traits, but in even the large testing programs, they were undersampling the frequencies of the stress environments, even with very large data sets. So we could have data sets, depending on the stage of testing, from tens to thousands of locations. But if you're continually sampling the wrong sort of part of the distribution, you've got 1,000 locations that don't give you a lot of information about some of the on-farm environments. So that, I mean, I spent a lot of time with the farmers in the western part of the US Corn Belt, and we just had to rethink how we were actually running our testing system to represent then their on-farm limited irrigation system. And it wasn't doing a lot of testing in high irrigation situations in the western part of the Corn Belt or high rainfall environments in the central part of the Corn Belt. And this allowed us to put more selection pressure on the traits and actually develop some of these products with improved stability and remove some of the products that had weaknesses that were sort of resulting in poor performance on farm. So it's not just size of testing system and numbers, it's really getting some key measurements of what are the conditions that we're sampling and how frequently do these occur on farm. I think both you and Gary are saying the same thing. I think we just got to start to rethink, how do we get information to allow us to know what the level of correlation is, and when it's low, how do we improve it? Because, I mean, basically, whenever I've found it's low, we've just had to work on new strategies for how to improve this. And that's going to depend on the system and where you're starting from. I mean, I didn't even talk about the rice work that I was involved in, but Gary's really familiar with this. And that's, I mean, we had a strategy of, instead of doing a lot of intense selection on single stations, we forced the breeders to do cross-testing across stations. It wasn't ideal, but it was at least a way of them getting a better understanding of G by E interaction, which they didn't think existed until they started doing testing across stations. So, I mean, I think you got to start from where you are and think about how to estimate this and how to improve it. And so, Eduardo, I look forward to the opportunity to discuss it further with you when I'm at Erie for the BPAT review. And it's the same discussion that I had with the CIMMYT breeders when I was in Mexico. The same topic came up and we spent a lot of time discussing this. So Gary talking about sparse testing is one way of implementing this. So if you can get your sampling strategy with sparse testing, it's a framework that could be applied to do this if you can get the sampling of the TPE adequately done.
Yep.
And it's an approach that I've used as well. It works really well as long as you're confident you're sampling the right aspects of the TPE.
Yeah, thank you, Mark. I think we are a little bit over time and it's quite late for Mark. There are several interesting questions and comments in the chat, which I might email to you, Mark, and you might offer some responses. Okay. Filippo, you lowered your hand. I wanted to give you the last time.
You said it's late, so.
Okay, all right. Thank you very much, Mark, for that interesting, and thank you everyone for the interesting discussion that has followed. For those who have questions in chat, I will have those emailed to Mark, and then we will have his responses.
And
back
Dorcas, I sent you the slides. You can send those around to everybody, and feel free if there's something that's not real clear with what I've actually sort of compiled in those slides, just you can send other questions as well. I'll do my best to try and answer them. Okay.
Yeah, thank you very much, Mark. Thank you.
Okay, thanks everybody.
Yeah, bye.
Bye.
Thanks, Mark. Thanks,
Hey,
Dorcas.
thanks,
Bye.
Michael.
Bye, Michael.
Thank you.
Bye.
Hi, Lisa. Okay. All right.